Okay, so I'm Alex Griffith.
I'm
That's
Katie Jordan.
Dr. K is our mentor, and we're with the Curcumin Project. So I'm going to start by explaining what turmeric is. Turmeric is a root or a rhizome, kind of like ginger. It's in the ginger family, and it's commonly used in foods in South Asia and Eastern Asia. And some people have even hypothesized that it could have been used for so long because of its antibacterial qualities, which we're going to get into later. And its yellow-orange color is due to curcumin, which is what I'm going to tell you more about right now. So curcumin comes in three forms, or curcuminoids, if you will. So there's curcumin, dimethoxycurcumin, and bis-dimethoxycurcumin. The difference between them is this one has two methoxy groups, this one only has one as a loss, and this one has zero. It's a loss of each one each time. So our research is interesting and unique because we're testing them all separately instead of as a group like people usually test them. So curcumin has this process called tautomerization, where one of these carbonyl groups will turn into an alcohol, and it's sort of a 1-to-1 ratio. But when it's in this form, it does something interesting which is important to us. So this is very polar. So the curcumin forms a chelate complex with iron. So chelating means it kind of grabs, it's like a claw. It grabs onto the metal ions, and that's very useful. So we're finding which curcuminoid is going to be the best at chelating iron. So in terms of testing, one method we're using is TLC, thin layer chromatography. It's sort of a preliminary method. So using TLC with a turmeric extract, so not a commercially made curcumin product, but directly from the plant, we found all three curcuminoids in great abundance. This is actually a really exciting plate. If you don't know about TLC, this is a really good plate. It's really like perfect separation. So it shows separation of curcuminoids, and it's a good preliminary technique to see what changes are happening to a solution. So we're also relying heavily on UV, ultraviolet spectroscopy. This is a little messy, I know. It's a lot of layers, so each line is a different test. It's great for colored solutions like curcumin, which is very yellow, which absorbs at about 425 nanometers, which is about there. But when you add iron, you get this bump here at about 530 nanometers, and that's really cool. That means a complex is forming.
Means what? Okay.
A complex is forming, so that's what it's showing. The iron also shifts this peak from 425 to 440. So when we tested different ratios of curcumin to iron, we made this graph, which I won't explain what it means, but basically it's different ratios of the ferric ion to curcumin complex. And this shows us that it's a 1-to-1 ratio of curcumin to iron 3. This is not to iron, this is 3.
2-to-1 ratio.
Yeah, 2-to-1 for iron 2. It's 1-to-1 for iron 3, which is another interesting thing.
Okay.
But we also found, we did a number of these graphs, and we also found that there's a second complex, which no papers are addressing, which is very interesting. There's a 1-to-1 ratio for iron 3, but there's also a 3-to-1 ratio. And then iron 2 and iron 3 are different. So there's a lot to research, there's a lot of data, and it's not very visually exciting, and I apologize. So another method we use is high performance liquid chromatography. So this is used to separate different compounds that are in a solution by measuring their polarity with a column. It's a little technical, the machine is very complicated, but that's the gist of it. So it shows, this is important because it can show a new compound. It's created in a solution, even if you can't physically see any changes. It's really exciting, actually. So in this graph, you can see all three curcuminoids, bis-dimethoxy, dimethoxy, and curcumin. Super cool. And this is before, and this is after the addition of iron. Once we add iron, these peaks sort of collapse into two. This is the free iron, and this is the new complex. So we see the new complex, and we have definitive proof that it exists.
Okay. So another method or test that we ran was infrared spectroscopy, or IR spectroscopy. And it's the analysis of infrared light interacting with the atoms of a molecule. And it enables us to determine the functional groups that are present within a molecule by measuring the vibration of its atoms. A frequency that a molecule absorbs at is called its resonant frequency. And the resonant frequencies are specific or kind of independent to each molecule. So that's what, or each functional group, excuse me. So that's what enables us to recognize what functional groups are present. The functional groups that are present in curcumin, we have 4. We have the benzene groups, which are the benzene rings. We have phenol groups, which is the benzene ring plus the alcohol. We have the alcohol group in the enol form that Alex showed you before, and then I said we have the keto group, which is these right here. So if you look up here, we circled this because that was one of our really exciting findings. The circle, the indication is actually where you find the keto carbonyl group, and this was before, and then once the iron was added, we actually lost that keto group. So you're seeing the loss of it, which was exciting for us. Yeah, so that's that, and then we have micro. So we created these agar plates, and we had 4 separate sections, as you can see. The first was the control group. The second and third, were iron and curcumin complex, but the iron was higher in the iron curcumin concentration. Sorry, was higher in 2, and then 4 was the same concentration of curcumin, but it didn't contain iron. So I know the picture might look a little blurry, but the zone of inhibition is larger for 2 and 3, and the zone of inhibition is where you don't see any of the bacteria. So the zone is bigger for 2 and 3 because it does contain the iron curcumin complex, and it furthers that point because 3 and 4, again, both have the same concentration of curcumin, but 3 contains the iron. So the complex is what is increasing the inhibition. So far, as you can see here, we have shown benefits thus far of curcumin. They're anti-tumor, antioxidant, antimicrobial, and anti-protozoal, which is really exciting. But we've also, most notably, it's being recognized for its anti-inflammatory properties, especially with arthritis. And when curcumin is combined with black pepper, it actually increases the curcumin's bioavailability, which is really great. If anybody has arthritis or knows anybody, curcumin and black pepper. But we're also, as you can see, looking into the antibacterial properties, and that's kind of what we're focusing on now in terms of the complex.
Thank you.